Hi, my name is Shannon Welch. I'm a junior in environmental studies at the Massachusetts College of Liberal Arts in North Adams, Massachusetts. Today, I wanted to tell you a little bit about my undergraduate research project, which is examining the plumage coloration of the dark-eyed junco. The slate-colored dark-eyed junco is a subspecies of the Junco hyemalis species. Slate-colored dark-eyed juncos are generally gray on the head and back with a white breast. They're a small songbird that's widespread throughout North America, with the slate-colored subspecies ranging from northern Canada to the northern United States, as you can see in the map in Figure 1. Slate-colored junco color often varies in subtle ways by age and sex of individuals. This means that those plumage color characteristics can often help distinguish males from females and younger birds from older ones. For example, females and young hatchier males tend to have more brown throughout their plumage than older males do. One characteristic in particular that seems to be consistent between young and old birds is the brown coloration in the edging of the tertials. Tertial feathers, as you can see in the photo on the bottom right of the poster, are the flight feathers that are closest to the body on the wing. Adult or after second year juncos will go through a complete molt in the fall months in preparation for migration. At this time, they will slowly replace all of their feathers, including their body feathers and their flight feathers. Some earlier literature supported the idea that brown-edged tertials were a reliable trait indicating a younger bird, specifically a hatchier individual in the fall or second-year individual in the later winter through the breeding season. Then, those individuals would be expected to replace their browner feathers during their first complete molt and replace them with a slate-colored feather, signaling an older individual. However, based on our earlier observations, we have found older banded males that still have this brown edging on the tertials. Further, some of these older males also show a larger amount of brown in the back and the head. This led us to wonder about this variation in degree of brown coloration in the slate-colored junco. While our long-range research goal is to understand the causes of this color variation, our objective here was to develop methods to describe and quantify the plumage coloration in slate-colored juncos. In order to first describe color variation in the slate-colored juncos, we initiated two approaches. First, in order to specifically address the color variation in the tertials, we collected one tertial feather from each bird we caught. We then took the tertial feathers to the lab and took photographs of them under standardized conditions, specifically with a black background and standardized lighting to eliminate shadows and get the best possible representation of the colors in the feather. We are still developing these methods and will not be presenting any of these results in this presentation. For our second approach to describing plumage coloration, we used a reflectance spectrophotometer. We were able to obtain a low-cost unit that's called the Spectra 1, manufactured by the Color Muse company. It sells for about $300. It's a small handheld unit that takes color measurements across the visible range of wavelengths, specifically 400 to 700 nanometers. We use this device in the field to take measurements across the junco's plumage. To take the spectrophotometer measurements, we gently held the unit against the bird's plumage in several locations on the body. The unit provided measurements of hue, value, and saturation, as well as reflectance at 10 nanometer intervals and between 400 and 700 nanometers. The hue describes the wavelength of the color. For example, blue is 500 nanometers and red is 700. Browns, which are dark orange in hue, fall between 590 nanometers and 650 nanometers. The saturation describes the intensity of this hue. Brown colors arise from an orange hue that is desaturated. The value describes the lightness or darkness. For example, very high values with low saturation appears white, for example, the belly color on juncos. Low values are black, which can mask any hue. This is what we find on the darker charcoal-colored juncos. Our interest is primarily in the degree of brown present in the plumage, which appears most noticeable on the head and the back of juncos. We determined the brightness over the brown wavelengths, 590 nanometers to 650 nanometers, on the head, top of the back, middle of the back, and rump for each junco. Using the program R, we tested for differences in these color metrics based on sex. Results showed significant differences in mean brightness over the brown wavelengths for males and females on some body regions. Females were brighter on their heads, backs, and rumps versus males. Based on the range of saturation values in our measurements, this means that females are measurably browner than males. We did not compare individuals based on age because we only had 22 birds in the analysis, and we lacked enough individuals in each age and sex class. While we have been able to demonstrate plumage color differences by sex, we are still working to determine how to best describe the overall plumage coloration of individual juncos. Visually, the differences in brown on some older male juncos versus others are obvious, but we are still not entirely certain how to quantify these differences. Finalizing these color methods are an important step to assessing possible drivers or correlates of that color variation. For example, during our bird sampling, we are also collecting a feather for measuring isotopes. These values will give us an approximate location of breeding areas and allow us to test for a relationship between degree of brown and breeding origin. We suspect that the intergrade zone between the slate-colored juncos and the Oregon juncos could be the source of brown heads and backs in the slate-colored juncos. Oregon juncos are very brown in their plumage, so interbreeding may cause morphological differences that may account for the variation in color among the individuals we're seeing. Thank you for viewing my presentation.